Hi, Shweta. Hello.
Hi, ma'am. Good afternoon.
Good afternoon.
Ma'am, there is an echo in your voice.
Is this fine, ma'am?
Yes, ma'am, it's fine, ma'am.
Okay. So I hope it's fine now. There is no echo.
No, ma'am, it's good, ma'am.
So okay, let's start the session. Just a minute. Shweta, you can stop me in between anywhere if my voice is lost because I'm in a less network area and the sound is also echoing. So if it is not audible properly, you can stop me in between or drop a message in the chat box so that I can get
Okay,
notified
ma'am.
about it.
Okay,
Okay.
ma'am.
Okay, thank you. So let's start today's session. My name is Kajal Kachava. I'm PMR Research Scholar from IIT Beju Varanasi. I will be your teaching assistant for this particular course of host-pathogen interaction, that is immunology, from January to April 2025. And today we are at the week 8 of this particular course, which is of total 12-week course. And until the week 7, the lecture material from my site is available on YouTube channel. You can check it out there. Also, the presentation is shared in your NPTEL folder. And my timing is every Saturday evening 3 to 5 p.m. So you can join me at this time. And the lecture material from Professor Himanshu Kumar sir is available until week 9 on the NPTEL portal along with its week assignment for the week 9. So I hope you might have checked it out from there. So today we will be solving the week 7 assignment, that is innate immunity and the complement system, on also about the adaptive immunity. So in the very previous slides of the initial classes where we have studied about history and development of immunology, there, if you remember, though, when we have studied this fever phenomena and some of the milestones of the immunology, where we have seen that the scientists told that some components or some proteins or some factors are responsible for the killing of pathogens. They were not mentioned any name regarding this. So I hope you remember this experiment of this fever phenomena where we have studied about this Vibrio cholera.
Yes, ma'am.
So briefly, what was in this experiment? What the scientists fever have done in this experiment and what was their conclusion from the study? Can you summarize very quickly if you remember, or should I summarize?
Yes, ma'am, I summarize it. There was an experiment carried out by Pfizer where mice were taken and few mouse were taken and it was injected with the Vibrio cholerae one, like Vibrio cholerae. Uh, and then when it was affected by the original pathogen, there was no infection. When the serum of the when the serum of the injected one is taken and subjected to the other animal, which is not being given any dose of, which is a non-immunized guinea pig, uh, there was some bacteriolysis that happened. So he concluded that there were some phagocytic cells that were present in the peritoneal cavity, and those caused bacteriolysis.
Yes, absolutely correct. So for a quick summary, what he have done that he have taken the Vibrio cholera bacteria and injected into the peritoneal cavity of non-immune patients. It was the group 1 that was non-immune animals, and he noticed that Vibrio cholera caused infection in those animals. In second case, what he did, he have taken the immunized animals, like who have underwent the Vibrio cholera infection recently and they were just recovered from the infection. So in the second case, what he observed that the bacterial destruction occurred without any cellular activity. That is, the within the 20 minutes, the Vibrio cholera bacteria was disappeared from that particular animals, and there was no phagocytic activity in that. So, what conclusion? And again, for confirmation, in the case 3, what he did, he have taken the serum from the immunized animals into the test tubes, and he have mix the Vibrio cholera into the test tube containing serum from immunized animals. Then what he observed that again the bacteriolysis occurred in the same way as in the case 2, that is in the case of immunized animals. So he have given a theory that this proved that bacteriolysis was due to antibodies and the complement proteins in the serum and not due to the immune cells. From here he told that no immune cells was involved, that is no phagocytic activity was involved. Perhaps the antibodies and some complement proteins were responsible for this bacteriolysis. Till then, the concept of this complement system and the action of antibodies was not confirmed. It was one of the initial theories regarding the complement system and the action of antibodies. So, from here, he gave a theory that in the serum there are some complement proteins and some effect of antibody which can cause the bacteriolysis. So, this was the first experiment. And the second experiment, we have studied about George Nuttall and Edward Buckner. What was in this experiment? What they did about George Nuttall and Edward Buckner.
It was an experiment where they used three different kinds of groups of mice, ma'am.
No, they have. This was the experiment with the clotting factor of the blood, like about
Uh,
some heating at some
Electrolyte
55
factors.
degrees Celsius. Then he observed that, okay, no problem, I will summarize this. What George Nuttall have done, his finding was that blood has inherent bactericidal power. From where it came till then, he was not aware about this. So what he have taken the defibrinated blood, that is the blood without fibrin, the clotting factor, and it was found that it was responsible for killing anthrax bacteria. How? What he have done that when blood was heated to 55 degrees Celsius, it lost its bactericidal activity. That is, the bactericidal activity was lost from the blood. So from there, he thought that there are some heat sensitive factors or proteins in the blood which are responsible for killing bacteria, and this indicated that blood contained natural antimicrobial compounds, which later known as the complement protein that we are studying right now, and some antimicrobial peptides. So, on heating at 55 degrees Celsius, bactericidal activity was lost. Otherwise, there was some inherent bactericidal activity in the blood. So, from this experiment, he suggested that the presence of some heat sensitive protein in the blood is responsible for providing immunity. Later, it was known as the complement protein. So, it was the experiment of George Nuttall. Similar experiment was done by Edward Buckner. What he told that he identified a bacterial killing factor in serum and called it as alexin. So Buckner discovered that blood serum, that is the fluid portion of the blood without cells or the clotting factor, could destroy bacteria. At that time, he named this factor as alexin, which later known as the complement system. I'm sorry. So this complement system consists of proteins that enhance immune responses, helping antibodies and phagocytes destroy bacteria and infected cells. So at that time, he thought that there is some factor in the serum, and he named it as alexin because at that time no theory about complement system was proven. So he confirmed that immunity could be mediated by the soluble factors in the blood. That is the humoral immunity. Like at that time, if you remember, then there were two theories about immunology. One was cellular theory, other was humoral theory. Some people supported the immune cells are responsible for providing immunity, and some people supported that no, there are some soluble factors or some antibody complement proteins related things are responsible for providing immunity. So there was a debate between humoral and cell mediated theory. So if you remember the initial slides, there were so many scientists. Some were supporting the cellular theory, some were supporting the humoral theory. So Edward Buckner was one of them who supported the humoral immunity, and he told that there are some soluble factors in the blood which are responsible for bacterial killing. So these were two experiments from Edward Buckner and George Nuttall. So from there, the concept of complement system was started. And later on in 1890, Julius Bordet at Institute of Pasteur discovered as a heat labile substance in normal plasma whose activity could complement the bactericidal activity of immune sera. Like he also told that there is some heat labile substance, that is something heat sensitive in the plasma that is responsible for providing immunity. So he have taken this like this theory, that is the sheep antiserum caused lysis of Vibrio cholera, that experiment, and other experiment was heating of the antiserum lost the lytic activity, and yes, lytic activity restored when the heated serum was supplemented with the fresh serum. That is, this fresh serum will also contain the complement. So, by the support of these previous theories, he have given his theory, Julius Bordet. So, he started the concept of complement system, and he told that a heat sensitive component is important for antibacterial or the lysis activity. And then later on, Paul Ehrlich also conducted the similar experiments, and he coined the term complement. So if it like, there were so many initial theories regarding complement, but Paul Ehrlich was one of them who coined this term complement, that heat soluble factors or whatever the complement proteins are present, their name is complement. But the term was complement by Paul Ehrlich. And later on, the soluble proteins and glycoproteins, there were so many experiments and theories related to this. So, what they have done here, they told that these are synthesized mainly by the liver hepatocyte and some other cell types. And it contained 5% of serum globulins, which circulate as inactive pro-enzyme, that is a zymosins, and they have the prolytic cleavage activity to remove the inhibitory fragment and it exposes the active site. Like they told this about the complement proteins, like what are these complement proteins consist of. And then the came what will happen if this complement is activated. Like, what is the role? Yes, till here we know that yes, there are some proteins, there are some complement factors responsible for providing immunity. So what? So what will be their action? So their first action is this cell lysis. That is the lysis activity. What will happen? These complement proteins will come and bind to this bacterial cell. That is the it can be bacterial, virus, or any type of pathogen. Let's say it's pathogen. That is the target cell. It will come and bind to the cell membrane here, and it will form like after binding, it will form pores in the membrane. Like we will study the pathways in the upcoming slides. So it will form here pores and the cell will be lysed. The cell death will happen. And the second case is of opsonization. What is this opsonization?
It is a process in which the target cells will be coated by certain molecules so that it will be easily recognized by our molecules to get phagocytosed.
Yes, so opsonization is like coating of pathogens to enhance the phagocytic activity. Like these factors will form surrounding and it will cause coating of this pathogen. So that like they will put some anchors or some flags on the this pathogenic cell so that it is ready for phagocytosis. So it is the opsonization. And the third case is of activation of inflammatory response. What will happen in this case? What will happen in this inflammation?
There are many steps that will occur during an inflammation where first uh, the capillaries will get enlarged to enhance the blood flow in the specific region of infection. Ma'am, are you asking about the yes, ma'am.
Someone is there, I think. Yes, please continue. What will happen here in the case of inflammatory response? Like if inflammatory response is activated, then what will happen because of that to the pathogen?
Um, Inflammation will occur, and that will inflammation will occur in such a way that the capillaries will be enlarged to increase the blood flow, and then it will recruit other phagocytic cells in the region of inflammation.
Yes, we will study again in the detail of when we will be studying about these pathways of the complement system. So, what will happen in the case of this complement activation? This pathogenic cell will be uh, represented by or will be tagged by some of the complement proteins, which will ultimately lead to the activation of the immune cells, which cells will cause the degranulation. We have studied so many times about this, which are the main cells responsible for containing granules. These are mast cells, basophils. These cells contains granules of the histamine. So these immune cells will release the histamine, which will increase the vascular permeability of the blood vessels. And also the other immune cells like this neutrophil and monocytes, every immune cell will come into action. neutrophil monocyte. Like these immune cells will get activated and they all will combine together and lead to the killing of this pathogen. So this will be because of the activation of this complement system. So there are like three ways of killing the pathogen. First one is direct lysis of this pathogen by binding of this complement protein to the target cell and causing the pores in the cell membrane of this target of the pathogen so that it will be killed. The second one is opsonization, that is this complement proteins will be tagged onto the pathogenic cell and this will undergo phagocytosis. So second way of killing is by opsonization. Third way is by the activation of inflammatory response. Like other immune cells will get activated and they will start the inflammatory response for pathogen killing. So these are the three ways of pathogen killing because of the complement activation. And then there's a nomenclature of complement. Like if you have studied or watched the lecture videos of this complement system, then you might be aware or a little bit familiar with these terms. There are some complement proteins like C1, C2, C3, C5, and also C3A, C3B, C5A. Like there are this so many complex and proteins. So what is their nomenclature designates for? It can be because with the numerals, that is the C1 to C9, and it can be with the letters, that is the factor D, and it can also have some names like homologous restriction factor. It is a name of a factor. So it can be either numerals, letter symbols, or some tribal names. And what they represents, like in the when you will study about this pathways of complement, like here you can see it will have like some C4, C3, A, B. You can see these things. My voice is clear and audible, no?
Yes, ma'am.
Okay. Yeah. So there are some peptide fragments which are made after the activation. Like one factor will break down into two factors. One will be containing A, one will be B. That is, A stands for the smaller fragment, B stands for the larger fragment. This we will study in the pathway. And there some complexes name is written with the bar on the top, like there is a bar above this name. So that is the complexes with enzymatic activity have bar on the top. So this is the name of a factor, that is the C4B2A. So this is the name of a factor and it contains bar on the top. It means it have enzymatic activity. So this is the nomenclature. So don't get confused with this. These are the names of the complement proteins. There are three main complement pathways. First one is this lectin pathway, second one is the classical pathway, and third one is the alternative pathway. And all together, their three ultimate aim is to kill the pathogen. So, what is common in between them and what is difference in between them and what you know about these three pathways in short, in general. I'm not asking the details, what is happening in each and every pathway. What are these three pathways about? Do you know that?
Yes, ma'am, a little bit. In
Yes.
classical pathway, it is activated by the immunoglobulins. That is the antigen-antibody interaction. And
Yes.
in alternative pathway, mm, uh, in alternative pathway, it is kind of a little bit complicated that involves different factors like factor D and factor B. And lectin pathway is activated by the mannose binding lectin, MDL. And their final aim is to produce something called a C3 convertase, which will ultimately give rise to another component called a C5 convertase, and all together will produce um like perforation membrane. What is that called? MAC. It's
Membranotic
called MAC.
complex.
It's membrane attack complex, and finally the cell will be phagocytosterialized.
Okay. Okay, correct. So what happens in these three pathways that as the name suggests, first one is lectin pathway. That is the mannose binding lectin, MBL. In the previous lectures, we have studied about some components of the this pathogen, something were there in some receptors we were studying in the previous slides. Can you recall some of the name of those? There also we have studied the similar things like on the
Proteins
pathogenic.
or receptors, ma'am.
Which one?
The proteins or the like the palms or the receptors, ma'am.
The pumps on the like pathogen associated molecular patterns. On the pathogen surface, there were some molecules we have studied similarly. I think mannose binding lectin we have studied there also.
Yes,
That
ma'am.
those were responsible for like suppose this is your pathogen. It contains some molecules on the surface, and these are
Lipopolysaccharides.
yeah, lipopolysaccharide that was in gram negative and gram positive bacteria, LPS and thicoic acid were present there.
Thicoid acid.
And also some of these lectins name were also there on in the PAMP only, that pathogen associated molecular patterns PAMP. Like these were recognized by the toll-like receptors. These PAMP, this is like on surface. These are the PAMPs, and these were recognized by this receptors TLR, NLR. These receptors were recognized by these. So here are the similar case, like these mannose binding lectin are present on the surface of these pathogens. Like these are the carbohydrates on the pathogen surface. These are recognized by our bodies, these complement proteins, like complement proteins in our body. And these MBL are present on the pathogen. Can you link the story between these both? Like these are present on the pathogen surface.
Yes,
MBL
ma'am.
is present here. It is being recognized by the complement proteins. Which complement protein? These are different in all the three pathways. Like in the lectin pathway, there are C2 and C4. And in this classical pathway, the C1 is also involved. And in alternative pathway, some factors are there. The every concept is similar. Like there are some soluble factors in the blood or the serum, which are responsible for recognizing the pathogen and their killing. So this is a overall summary of this complement pathways, that in lectin pathway, mannose binding lectin will be recognized by our immune system. And in classical pathway, there will be antigen and antibody interaction. Like in this classical pathway, antigen and antibody interaction will happen, and the antigen antibody complex will be formed. And in the alternative pathway, these factors are involved, and it is antibody will not be involved here. It is antibody independent pathway. It is antibody independent alternative pathway. So, and their ultimate aim is to form the C3 convertase, which will cleave the C3 and it will form C3A, C3B, which will ultimately bind to some other factors and it will form the membrane attack complex and it will cause the cell lysis. So this is in general. We will study each and every pathway in detail. So these are the pathways. The first one we have studied here is lectin pathway. The second one is here classical pathway, and the third one is here this alternative pathway. So uh, is this slide complicated right now, or you can understand everything clearly from this, like what is happening? The three
No,
pathways
that
are
is
clubbed together
understandable.
here. It
Yes,
is
ma'am, it is
understandable.
understandable because I watched the video.
Yes, yes. So if you watch the video or if you get the concept of what is happening in this complement pathway, then it is very easy. There is no need to get confused between all the three pathways. But for a first time who have not heard this name before, for them it might seem complicated. So don't get worried about this. We will cover each and every pathway separately. Then you can understand easily this one slide of all the three pathways together. So this is also the same pathway in different form, like there are three pathways here also. This you can, sorry. So here also this first one is lectin pathway. I think result. So okay, you can get it like the first one is here lectin pathway, second one is classical, third one is alternative pathway here. So if you start with the classical pathway, so this is antibody dependent. Like in the classical pathway, antigen antibody complex will form. And the common antibodies which are mainly involved in the classical pathway of complement system are IgM and IgG. And in early stages, the C1, C2, C3, and C4, these four complexes are involved from the complement. So, what you understood from this particular slide, the structure of this complement protein?
There are three different domains. One is C1Q, where and there are two other domains called the C1R and S, which are like they have two molecules of C1R and S, and the C1Q is the main region which will get attached to the antigen antibody complex, and it is like a collagen triple helix where collagen is rich in proline, and it is a collagen like protein. There is a triple helix in the staff like region that will bind with the antibody molecule.
Yes. So, like in this classical pathway, the C1 complex is the initiator of this classical pathway of complement system. Like C1 is the initiator complex. So remember this, that C1 is the initiator complex of this classical pathway of complement system. And the structure of the C1 complex is like it have the C1Q, which is the six globular head connected by this collagen triple like helix, like these six stalks are connected. So this is C1Q. And then it have the C1S and C1R. So the C1R is an inactive serine protease that gets activated upon C1Q binding. So remember this, we have studied in the previous slide about the zymosins, that is the inactive form. Like the C1R is currently inactive. It will get activated on the binding of the C1Q. And this activated C1R will cleave the C1S and it will activate it to proceed with binding to the like cleavage of C2 and C4. Understand it like this. C1Q is first person. For here, I'm saying this first person, but these are complement proteins. So, like, understand it, C1Q is first protein, C1R is second, C1S is third. So, the C1Q is the main. It is the this six globular head. In this have structure of the six. This is a six globular head connected by collagen triple helix. So this is the story of C1Q. And the C1R is, it is inactive serine protease. It will get activated on the, it will get activated after binding with this C1q. Once C1r is bound with the C1q, it will get activated. It will get activated. And once the C1r is active, it will cleave the C1s. Like the C1r, once it is active, it will cleave C1s. And this will, C1s will again proceed for the cleavage of next factors, that is the C2, C4, and this C2 and C4 activated will proceed further for something else. So the story of this C1 is a complex, C1 is a protein of this complement system, or it is a complex, C1 complex. It have three subunits, Q, R, S. Q is six globular head connected by collagen triple helix. C1R is second subunit, which is currently inactive serine protease. It will get activated on the binding of the C1Q. Once the C1R is active, it will cleave the C1S, and the C1S activated, it will proceed to the cleavage of C2, C4, and so on the downstream pathway. So you understood how these three subunits are connected together.
Yes, ma'am.
So this is the complete pathway, that is the classical pathway. This is the classical pathway, and these are the five steps we will take one by one. The first step is that C1q binds antigen bound antibody. Like on once the antigen is entered into any like host cell, then antibody will bind to that, and the C1q will go and bind to the antigen antibody complex. And this C1R activates autocatalytically and activates the second C1R, and both activates the C1S. Like that we have seen in the just like last slide, that this antigen antibody complex will be formed, and the C1q will go and bind to the antigen antibody complex, which will activate the C1R, and C1R will again activate the C1S. Antigen antibody complex will bind with C1q. C1q will activate C1R. C1R will activate C1S. So this is the pathway here. Then the from here what happened? We have the activated C1S. So in second step, what will happen, that the C1S will cleave the C2 and C4. And this cleaving C4 exposes the binding site for C2, and the C4 binds the surface near C1, and C2 binds C4. And these will form the C3 convertase. So what is happening here? We have this activated C1S. It will cleave C4 and C2. And this cleaving C4 will expose the binding site for C2, and the C4 will bind to the surface near the C1 and C2, and they will form C3 convertase. You will see so many terms like C3 convertase, C5 convertase. So keep in mind that what is the sequence of pathways happening, like who is activating whom. So here this C4b will bind with or will club with this C2A, and they both are known as the C3 convertase. So from here we have the C3 convertase. In the next step, what will happen? The C3 convertase will hydrolyze many C3 molecules, and some will combine with C3 convertase to form C5 convertase. Like what will happen here? Now we have the C3 convertase. And who is this C3 convertase? It is C4b combined with C2a. This is your C3 convertase. See, from here we have this. This is C4B2A. So the C3 convertase, what it will do, it will ultimately hydrolyze so many C3 molecules, and some of those C3 will bind to this complex. That is, we already have the C4B2A, and some of the C3B molecules will bind with this, and now this complex is called the C5 convertase. You understanding what is happening here? Like one molecule is hydrolyzing the next subsequent molecule and it is getting activated, and that is again combining with some downstream molecule and forming a next molecule. Like from starting we had the C4, then C2B combined with this C2BA, then again finally the C3B combined with this, and it formed the C5 convertase in the step 3. And this C3B component of C5 convertase binds with the C5, permitting C2B2A to cleave the C5. Like what we have from here, C4B, 2A, 3B, C4A, sorry, 2A. So we have this. This is your C5 convertase. And the C3B component of C5 convertase, it will bind with the C5. It will bind with the C5 and it will permit this, the remaining C4B2A, to cleave the C5. Like this is a long molecule, C4B, 2A, 3B. So this C3B component of this molecule will bind with C5 and it will allow this remaining molecule to cleave the C5. Like here the C5 will be cleaved, and the C5 will be cleaved into what? It will be cleaved into C5B and C5A. So from the last step we have here C5B and C5A. So the C5B, it will bind with C6 and it will initiate the formation of membrane attack complex. That is the MAG. This was their ultimate aim from the beginning, to finally form the membrane attack complex. It is present on the pathogen surface and it will cause the cell lysis and pathogen death. So what will happen here? From the previous complex we have C5B. Here the C5B, it will bind with this C6, and again they will form this, they will subsequently bind with the C7, C8, C9, and finally they will form the membrane attack complex on this pathogen surface, and it will be dead. So this is the whole pathway that we have seen from the initial. What was happening in the first step, the C1Q was binding with the antigen antibody complex and it was activating C1R and C1S. Then the C1S was activating C2 and C4, which were forming the C3 convertase. And the C3 convertase again bound with some molecules and formed the C5 convertase. And the C5 convertase again bound with the C6 and it initiates the formation of membrane attack complex, and it combined with other like the C6, C7, C8 and C9, and it ultimately formed this membrane attack complex which killed the pathogen. So this was the classical pathway of complement system. So I hope this is clear and easy to understand. Now there is no confusion in this.
As well as clear.
Okay. So you have to keep in mind that this pathway it started from here. Like this, C1 is the initiator of this classical pathway. And in the first step, the antigen antibody complex is involved in the classical pathway. These things you have to keep in mind. These are the main key points of this classical pathway. And ultimately, they form the C3 convertase, C5 convertase, and then the membrane attack complex. This is the how a membrane attack complex look like. Like the spores are formed in the cell membrane. So the ions and the small molecules can freely pass through the pores, and the cell cannot maintain the osmotic stability, and cell will get lysed. So this is a image. This is a photomicrograph of a pathogen surface. So you can see here that this pores are formed in the membrane. So like it is not possible for the survival. So cell will get dead. The second pathway is alternative pathway. So this is antibody independent pathway. Classical pathway was antigen antibody complex dependent pathway. Alternative pathway is antibody independent pathway. And this is a component of innate immune system because the antibody is not involved here. So like it will not fall under the adaptive immune system. So this is a part of innate immune system. And the early stages involve this C3, factor B, factor D, properdin, and it is initiated by the cell surface constituents which are foreign to the host, like for example, gram-negative and gram-positive bacteria. Like these bacterial surfaces act as a foreign particle for the host, and it will initiate the alternative pathway of complement system. So this is the complete pathway of this alternative pathway. This is your alternative pathway. What is happening here? The C3 hydrolyzes spontaneously and C3b fragments attaches to the foreign surface. Like the C3 will get hydrolyzed into C3a and C3b, and the C3b will attach to the foreign particle surface, and this factor B will bind to the C3a, exposes site attacked on by this factor D, and this cleavage generates C3b combined with factor B, which has C3 convertase activity. In this previous classical pathway, who has the C3 convertase activity, it was C4, the C4b to A. Here, the C3 convertase activity is present with this fragment, that is C3b combined with factor B, small subunit of this factor B. So what is happening here? The C3 is getting hydrolyzed into C3b and C3a, and the C3a is binding with this factor B, and it is exposing the site for the binding of this factor D, and this cleavage will generate this C3b and factor B and the small subunit of factor B. Then this properdin will come and bind to the C3 convertase. It will stabilize this C3 convertase, and then what will happen? This convertase will generate the C3b and some bind to C3 convertase activating C5 convertase. Like this properdin will come and bind to this C3 convertate, and it will stabilize this complex. Here this complex is formed of the C3 convertase, and this will activate the C5 convertase. And again, what we have seen that after the activation of the C5 convertase, the membrane attack complex will form, which will kill the pathogen. So similar thing is happening here. The concept is same of formation of C3 convertase and the C5 convertase, and finally the formation of this membrane attack complex, which is their ultimate aim of this complement system. But in this pathway, because the antigen antibody complex is not involved, it will directly go and bind to the pathogen surface. So some of the factors are different. So those name you have to keep in mind that in alternate pathway factor D and factor B are involved, and properdin is involved, which is responsible for the stabilization of C3 convertase. So these two three things are different in this pathway. So that you have to keep in mind. Otherwise, the concept is similar for this as well. So I hope this pathway is again clear. The alternative pathway.
Yes, ma'am.
Okay. If you have any doubt or any confusion, so you can ask. No problem.
Okay, ma'am.
So there are some initiators of the alternative pathway of this complement system. Like there are some pathogens and particles of microbial origin, like some strains of the gram-negative bacteria and this lipopolysaccharide from the gram-negative bacteria and some strains of the gram-positive bacteria and teichoic acid from the gram-positive cell wall. There are some fungal and yeast cell wall components like the zymosens, and there are some viruses and virus infected cells. There are some tumor cells like Raji cell. There are some parasites like trypanosomes, and there are also some non pathogens like human IgG, IgA, IgE, and rabbit and guinea pig IgG complex, cobra venom factor, heterologous erythrocytes of this mouse, rabbit, and chicken, anionic polymers like dextrin sulfate, and pure carbohydrates like agarose and inulin. These are some of the factors or the some of the initiators which act as a foreign particle for the host cell, and it activates the alternative pathway of this complement system. So there are some of the examples that you should keep in mind. The third pathway is your lectin pathway. So, what is peculiar about this lectin pathway? That it initiates by the binding of this mannose binding lectin. So, because that's why the name is lectin pathway. And it is also antibody independent. If the question is that which complement pathway is dependent on antibody or antigen antibody complex, then that is the classical pathway. Otherwise, this lectin and the alternative pathway both are antibody independent pathway. And lectin pathway uses C2 and C4 complex, whereas your alternative pathway uses this your C3 and factor B, D, and properdin. So these factors you should keep in mind, like C3 is involved in your alternative pathway and C2 and C4 are involved in your lectin pathway. And in classical pathway, all the this complex proteins are involved. So what is this mannose binding lectin? What are these? Where are they present?
They are present on yeast uh, wall, cell wall,
Mm,
and also present in some most of the prokaryotes, but not in the eukaryotes.
okay. Like these
They
are
act
the
as a terminal sugars.
terminal sugars of the on the cell membrane. These are the like glycoproteins or the carbohydrates present on the cell surface of these pathogens. So these are recognized by our immune system as foreign. So they will get bound to this complement proteins and the complement system will get activated. Like there are some sugars in the yeast and the vertebrates. You can check out their names from here, like this. Blue circles are your glucose, green circles are your mannose, this blue squares are your N-acetyl glucosamine, and this your red triangle is for sialic acid, yellow circle is for galactose, and this red triangle is for fucose. So these are the sugars or the carbohydrates present on the pathogen or like bacterial or yeast cell wall or cell membrane. So like this is simple, like this mannose binding lectin present on the microbial cell wall will uh, these will activate this C1 like complex, And again, it will follow similar pathway like the classical pathway. But classical pathway was getting started from your antigen antibody, whereas your lectin pathway will get started from this mannose binding lectin. So this is the major difference in that. And this MBL will activate this MASP, that is your MBL associated serine protease. And it will ultimately activate your C1-like complex. And again, the similar pathway will take place. And their final aim is to form the membranatic complex. All the three pathway will come here, and they will lead to the cell lysis. So after studying all these three pathways, now if you look at this one slide, then is it now easier to understand the pathways? Like first one is classical, second your lectin, third is your alternative pathway. So from here, in a single site, you can see that classical pathway is getting started from your antigen antibody complex, and C1 is the initiator complex, and the C1 will activate C2, which will activate C4 and C3, and this will form your the C5 convertase, which will activate your C6, 7, 8, 9, and they all will club together and form the membranotic complex. Like from here to here, this is your classical pathway. And from downside, if you see the alternative pathway, then this will get activated by your microbial surfaces. Microbial surface will act as a foreign to the host cell. So this will directly activate your this C3 complex, which will activate your factor B. Factor B will break down into factor BA and BB, which will bind with the factor D, and here it will somewhere here it will bind with this your properdin for stabilization, and the C3 convertase will form the C5 convertase, and after the C5 convertase formation, again it will bind to the C6, 7, 8, 9. So from here, your alternative pathway, like till here the C5 convertase is common, then again it will form this membranotic complex. So this will come and mix here with the after the C5 convertase formation. And same here if we go with the lectin pathway. So it will start with your mannose binding lectin, and it will involve your this MBL associated serine protease. Then it will activate your C1 complex. Then it will go to activation of this your C4, C2, and the C5. Then this will form your lectin pathway. So in the single slide, what you can observe the main points are classical pathways initiated by antigen antibody complex, alternative pathways initiated by microbial surfaces, lectin pathways initiated by mannose binding lectin, and they all will form in common is your C3 convertase or your C5 convertase. And after the C5 convertase formation, they will go and bind to C6, 7, 8, 9 complex, and they will form the membranotic complex, and it will fill your cell. So this is the common of all the three complement pathways. So I hope all these three pathways are clear and no confusion. If you have any doubt or any query, so you can ask.
It's very clear, ma'am. You put it well together.
Okay. So don't get confused between these. The uh, only the classical pathway is your. Wait, I will write it here somewhere. Classical pathway is your adaptive immunity. It involves your adaptive immunity because the antigen and antibody complex formation is involved. So it comes under your adaptive immunity. Second, your this alternative and lectin pathway. These both are your antibody independent pathway. That's why they will come under your innate immunity. This is the main point that you have to keep in mind. Got the point? How they are different in this scenario?
Yes, ma'am.
Okay. So now there is a regulation of this complement. So these complement components are capable of attacking the host cell, and these complement components undergo spontaneous inactivation if they are not stabilized with other components, as it is highly labile component. Like we have seen that one molecule is breaking down into subunit A and subunit B, which is again binding to some subsequent subunits, because so that they can get stabilized. Otherwise they are like highly labile, so they will get inactivated. So C3 convertase is major amplification step in all three pathways, therefore that needs regulation. Like starting from here, the C3 convertase is formed in all the three pathways. Like small subunits are clubbing together and forming the C3 convertase. So there are some proteins that regulate the complement system. Like I will share this presentation so you can go through this slide and see all these factors and which pathway they are involved in and what are their immunological function. So you can check it out from this table. And there are some biological effects which are mediated by this complement products, like cell lysis will happen after the formation of this membrane attack complex. And in membrane attack complex, the C5b and the C6, C7, C8, C9 complexes are involved. So there are some complement products which mediate the biological effect. So this table also you can check it out. Like here, for example, the degranulation of mast cells and basophils are because of which type of complement products? These are because of the C3A, C4A, and C5A. That is the A is smaller fragment or larger fragment?
A is a smaller fragment.
A is smaller fragment. So the smaller fragments of the C3, 4, and 5 are responsible for the degranulation of mast cells and basophils. That is, this phenomena is called as the anaphylotoxin. Have you heard this term before? Anaphylotoxin?
It was mentioned in the lecture, ma'am, but I don't know the meaning.
At what point or with what context it was mentioned? Like, if you can tell me so I can relate that and explain it to you.
It was said that in the A component, 5A component has anaphylotoxic effect, and this will be discussed later.
Okay. Like this anaphylotoxic are these factors, like these smaller fragment, this C3A, C4A, C5B, these are the smaller fragments which are responsible for activating your immune system and again uh, ultimately activating your this inflammatory response. Inflammatory response by your immune cells. So though at the initial slide somewhere I told you about this activation where it was inflammation. Like this third one, the activation of inflammatory response and the degranulation of immune cells, and this third mechanism, this is called the anaphylotoxin. This is here anaphylotoxin, and this degranulation and inflammatory response is generated because of your this complement subunits. I think I have included it somewhere or I missed to include it. But you understand this point, no? Now, what is this anaphylotoxin?
Yes, ma'am.
This is an inflammatory response by the immune cells. Like here, also this mentioned that degranulation of mast cells and basophils is because of these subunits. And the next one is this degranulation of your eosinophils is because of your this C3A, C5A. So you can check it out from this table. There are so many biological effects like this opsonization of this particulate antigens, increasing their phagocytosis is mediated by this subunits. Like these smaller subunits of your complement proteins mediates your degranulation, whereas your this larger subunits of your complement proteins mediates the opsonization. Smaller one mediates your degranulation. And this viral neutralization is mediated by your C3b and C5b and until your C6, 7, 8, 9 subunits. And the solubilization and clearance of your immune complex is mediated by the C3b. So you can check it out from this table. This is a similar table, like so your soluble factors will regulate your complement, how they will regulate, like which factor will bind to which factor and what will be their action and what will be their pathology. Like here, this your C1 inhibitor, it will bind to your ligand or binding factor is your C1R, C1S, C1Q. These are its binding factor, and its action is to displaces the C1RS and MASP2, inhibiting activation of the C1Q and MBL. And what is its pathology? That is your this hereditary angioedema. What is this? Your hereditary angioedema. Have you heard this term before?
Yes, ma'am. It is a condition where edema occurs in the muscles or something, ma'am. But there is an edema.
Yes, like there is a, severe, like life-threatening inflammation type of on the body,
Mm.
and it is a genetic disorder. It is very rare, but this is genetic disorder. Something like life-threatening inflammation type of condition happen in this case. So this is because of your the C1 inhibitor. So these are some pathological conditions that you should keep in mind. Like here, this factor H, which binds to C3b, and its action is to displace the this factor B, B subunit of this factor B and cofactor for factor 1. And its pathology is age-related muscular degeneration and atypical hemolytic uremic syndromes. So these are the pathological condition with your factor H. And your factor I is also somewhere involved in some mechanisms of the complement. It will bind to your C3b, C4b, and its action is serine proteases, which cleaves the C3b and C4b, and its pathology is low C3 levels and hemolytic uremic syndrome. So these are the pathological conditions because of this. Some of your complement factors or complement subunits. So this you can check it out from here, and this like this protein S, it is responsible for binding to your this complex and it will inhibit the MAC formation. Like if protein S is present in your in where somewhere between in your pathway, then it will bind to your this complex. That is your somewhere after the C5 convertase, and it will not like see here it will bind to like C6 and C7 only. So 8 and 9 will not bind here. 8 and 9 are not present here. So what will happen? It will not allow the formation of MAC complex, because as you know that in formation of your membrane attack complex, what you need your C5 convertase, that is C5b, plus you need your 6, 7, 8, 9, the remaining complement subunits. Then only your MAC will be formed. So if this protein S is present in your pathway, it will not allow the formation of MAC. So these are some of the factors. You can check it out later. And again, similar, there are some more factors and their action and their pathology. This is also similar. There are some microbial pathogens which invade your complement system. Like if there is your Neisseria meningitis, it what will do? It will bind with the factor H protein, and its host target is also factor H, and it will inactivate your the C3b unit. Like if your C3b is inactivated, C3 convertase will not form, C5 convertase will not form, your MAC will not form. Similarly, with the case of this Borrelia burgdorferi and Streptococcus pneumoniae, so these all will inactivate your the C3b. They will not allow the formation of your C3b, and hence your MAC will not form. Hence your pathogen will not be killed. So these are some of the membrane proteins and some of the secreted proteins. So this table also you can check it out. There are some complement deficiencies like deficiency of C1q, C1r, C1s, C4, C2. They will show the enhanced immune complex and show SLE like symptoms and glomerulonephritis, vasculitis, and the individuals also show recurrent infection by pyogenic, that is a pus forming bacteria. And this is your deficiency of this C1 complex. So, deficiency of your C1 complex and this C2 and C4. And deficiency of your factor D and troponin will show the Neisseria meningitis infection, but not with meningococcal bacteria. So these are some of the deficiencies you can learn from here. And this next deficiency is your deficiency in MBL, that is mannose binding lectin, show increase in respiratory tract infection. Deficiency of your C3 will show frequent severe bacterial infection. This protectin, which inhibits the C9, therefore it inhibits the MAC. Its deficiency will give a high risk of thrombosis. 6 one is your anti-C5 is used for the treatment of paroxysmal nocturnal hemoglobinia. This is a condition came in the name of this something something. So you can check it out from here. So there are some of these deficiencies of this complement protein which lead to some disease conditions. So next is your very main and very basic topic of the immunology, that is the innate and adaptive immunity, that we have already covered in very previous initial first or second lecture of our this particular course. So I will not go into much detail of this. So I think you have seen this from the lecture videos. If you have any doubt, you can ask. We can discuss this. Otherwise, I will quickly take this because we have to solve the questions as well.
Ma'am, a quick revision will do.
Okay, so like there are key cellular players of immunity. In adaptive immune system, there are B lymphocytes and T lymphocytes. In innate immune cells, there are immune cells like neutrophils, eosinophil, basophil, and natural killer cells. And there are some of the immune cells which bridge between the innate and the adaptive immunity, like they are involved in both the immune system, or they are uh, like they are acting as like antigen presenting cell, like they are taking like they are binding to the antigenic cell or the pathogen from the innate immune system, and they are calling the immune cells and the antibodies to act upon, like they are acting as a bridge between both innate and adaptive immune system. So their examples are macrophages and dendritic cells. These comes in both the systems of innate and adaptive. So there is a comparison of innate and adaptive immunity. So first one is their presence, like as we know from our 10th 12th standard definition that innate immunity comes by birth. Innate immunity is present in our body by the time of birth. So the first is their presence, that is the innate immunity is already present in the body, while the adaptive immunity is developed upon exposure to a foreign substance. Like when we are exposed to any pathogen, then our immune system will get activated. That is your adaptive immune system. And if we talk about specificity, then innate immunity have a broad specificity, and while the adaptive immunity is highly specific. Highly specific means it is very specific to a pathogen. Like antibodies are involved, they will go and bind to only the specific antigen present, while the broad specificity is like it is non-specific, it will act to all the pathogen in a similar way. So there is a difference between this both the specificity. So the third one is the response. That is the in innate immunity. It fight with any living or non-living foreign entities. And in the adaptive immunity, what happens that it develop highly specific immune response. It takes time and develop the immune system. So this is the response time. Response time is here rapid. It is like in innate immunity, it is minutes to hours. But in the case of this adaptive immunity, what will happen? There is a slow response. Like it can take some days to some weeks in taking the response towards any pathogen. And if you talk about the potency, that is what is the response like? You can say this the intensity of the response. That is innate immunity is similar, quite non-specific. So its response is limited or the potency is lower. While the adaptive immunity is very specific, the antibodies are formed to a particular antigen, so it is highly potent or it have a high intensity of response. If you talk about time span, then every time the response is generated, irrespective to the number of exposure. And in the case of this adaptive immunity, once immune response developed, the immunity can be lifelong or short, depending on the pathogen. So like this. This is the time span. The next one is your inheritance. The innate type of immunity is generally inherited from parents and passed to offsprings. Like if we say if a mother have a strong immune system or she have a high immunity, then probably the child will have a strong immune system and vice versa the case. But in the case of adaptive immunity, it is not passed from parents to offspring. Doesn't matter how your parents' immune system are, your immune system will develop on your exposure to the environment and how your immune system is developing adaptive immunity towards the particular infection. So it is not inherited. In the case of intensity of immune response, it cannot react with equal potency upon repeated exposure to the same pathogen. Like if we have a common cold, then our body sometimes you will notice that our body can recover within like 1 to 2 days. Sometimes it takes like around a week of your recovery. So it is not same all the time. But in the case of your adaptive system, it can remember the specific pathogen which have encountered before. So its intensity will increase over the your number of exposures. And like it is present from the birth, it develops during the lifespan, and it don't have any allergic reactions. And this adaptive immunity have immediate or delayed hypersensitivity reaction. This hypersensitivity you will study in somewhere upcoming topics. And it don't have any memory, but adaptive immune system have a long-term memory. And if we talk about the diversity of this immunity, then innate immunity is very limited, while adaptive immunity is high. Diversity is high. And complement system activation, we have just discussed about this, that alternative and your lectin pathways fall under your innate immunity, whereas your classical pathway fall under your adaptive immunity. Why? Because here antigen antibody complex was involved and these two were independent of the antibody. The anatomical and physiological barrier in innate immunity is skin, mucous membrane, temperature, pH, chemical. These act as the anatomical barrier, like they will not allow the pathogen entry into your body. But in the case of, if we talk about adaptive immunity, the anatomical or physiological barriers are your lymph nodes, spleen, mucosal associated lymphoid tissue. This we have studied in the organs of immune system, if you can recall this. So we have studied this here primary and secondary lymphoid organs and some of the lymphoid tissues. If we talk about the composition of this innate immune system, then it is composed of this physical and chemical barriers, phagocytic leukocytes, dendritic cells, NK cells, and some plasma proteins. But in the case of this adaptive immune system, it is only composed of B cells and T cells. It is very limited. And the next one is your development. So innate immunity is evolutionary, older, and found in both vertebrates and invertebrates. But adaptive immune system has developed recently and it is found only in the vertebrates. In invertebrates, there are no production of antibody and such kind of stuff. And the examples of your innate immunity is your WBC fighting with bacteria and causing redness and swelling when you have a cut. So this is an example that your WBC will fight with the infection. And if you have a cut, then you will have swelling and redness. That is the signs of your inflammation reaction. But the examples of your adaptive immunity are chickenpox vaccination so that we don't get chickenpox because adaptive immunity system has remembered the foreign body. That this we have studied in very initial lectures about the um, innate and adaptive. Then we have studied two terms, natural, artificial. Then we have studied active and passive. Do you remember these terms that we have studied in the initial, very initial slides?
Yes, ma'am, in the first class.
In the very first class, I think we have seen like one or
Yes,
two
ma'am.
slides along with its examples of what case
Yes,
is
ma'am.
the natural active, natural passive, active artificial, innate, and everything you have seen in that class. So I missed to put that slide here, but on sharing the presentation, I will again put that two slides here so that you can quickly revise it from here. So this is the very quick summary of your innate and adaptive immunity. And these are the key basic points that you should always have on your tongue when you talk about innate and adaptive. The first one is response time is minute to hours for innate immunity and for adaptive it is for days. Specificity is limited and very fixed and adaptive is highly diverse, adapts to improve during the course of immune response. The response to repeat infection is same for each time and it is more rapid and effective with each subsequent exposure. The major components are here your barriers, phagocytes, and some of the pattern recognition molecules. But in the case of adaptive immunity, these are T and B lymphocytes and some antigen specific receptors and formation of antibodies. So this is a quick revision of your innate and adaptive immunity. So I hope this is clear, this quick summary. You have recalled everything, whatever you have studied about innate and adaptive. So I think this is
Yes,
clear.
ma'am. Yes, ma'am, it's clear.
Okay. So the next one is your antigen and antibody. There is a separate portion in the upcoming slides or the, I think in the just next week session. Have you gone through the lecture videos of week 8? I think about antibody classification, diversity, I think it is mentioned there as much I remember. So we will take these things very much detailed in the upcoming classes. For a short uh, revision here, the antibody structure, it have light chain, it have two of the light chains and it have this two of the heavy chains. These inside one are your heavy chains, outside smaller one are your light chains. It have disulfide bonds between the two chains and it have interchain and intrachain disulfide bonds. It have here the antigen binding sites, that is here at the upper side. At the lower side, it is the your Fc portion. This is your Fc at the lower side, that is your fragment of crystallization. It has the effector activity or the biological activity. So we will take this in very much detail in the upcoming slides. So don't worry about this. Don't get confused with this complex structure. It is very simple and very easy to understand, as like the complement pathways. So don't get confused. I will cover this in the upcoming classes. So for now, just keep in mind there are five types of antibodies. Do you remember their names, or you have a problem in recalling their name every time you missed how many types of antibodies are there? There are five types.
Mama, recall them now.
Okay, like there, you can remember this like GAME, then D. Game, then D. You can remember it like this. I have remembered this from like my 12th standard of bachelor's like this, and IgG, IgA, IgM, IgE, and IgD. And this is very much easy to remember. Otherwise, you can have your own way of remembering, no problem with that. The most common found is IgG, and the biggest molecule in the size, that is pentameric, that is your IgM. And this IgA is your dimer, it contains J chain in between this. And this your IgE is your that anti-allergic hypersensitivity mediated antibody, that is your IgE, and IgD is occurring very rarely. So there are five type of antibodies. For now, you just remember this much. For the upcoming classes, we will cover more. So, so there are two terms. One is immunogenicity, second is antigenicity. What is their difference between that? What are immunogens and what are antigens? Do you know the exact difference between these immunogen and antigen?
No, ma'am.
You don't know about these immunogen and
No.
antigen.
I haven't gone through the 8th week lecture yet.
This is the week 7 on. Okay, means you have not
Yeah, this
submitted the
only
assignment.
for week 7 I have completed and submitted, ma'am. This part falls in week 8 actually.
Okay, in detail, it might be in week 8, but I think somewhere in lecture videos it was in week 7. That's why I have included this here.
That antigen antibody thing was in the week 7, ma'am, but this immunogenicity and antigenicity is in the week 8
Okay, means
portion.
this is in
This
week
we
8
can,
portion, so we can discuss it in more detail there.
okay, okay,
Okay,
ma'am.
okay, no
But
problem.
only one question is from this part.
Okay, no problem. There is a quick difference between these two terms. One is immunogen, other one is antigen. Like this like immunogen will activate your immune system. But for antigen, it is not necessary to trigger your immune system. Like immune system, it will always activate your immune system. Like immunogenicity, it induces immune response such as antibody production. But in antigenicity is or antigen is your not necessarily induce immune response, but it can induce immune response if appropriately modified. That is the heptanes. Like it can. It's not necessary that always an antigen will activate your immune system, but your immunogen will always activate your immune response. There is a line like there's a one sentence, all antigens are immunogens, but not all immunogens are antigens. Something like that. So you can write it somewhere and go through this. We will have a discussion on this in the next week about immunogen and antigen. I will explain it more clearly in the next week because it will take some time. For now, you just keep in mind that immunogens will induce, sorry, these will induce the immune system and it will lead to the production of antibody. But antigen not necessarily induce the immune response, but it can induce immune response if appropriately modified. That is, here comes the case of heptins. Okay, so you just keep in mind immunogen and antigen. Just write this. It will always activate the immune system and it is not necessary for antigens. So, what are heptanes? These are small molecules which lack the immunogenicity on itself, but these can be bound to the antigen or any carrier molecule, then they can induce the immune response. Like these are the small molecules. Itself alone, they will not induce our immune response. Just So, okay, these heptans are your carrier conjugates. These produce three types of antigenic determinants, that is, antibodies to heptane, antibodies to carrier, antibodies to heptane carrier conjugate. Like these, your heptanes are the small molecules. Itself, they are non immunogenic. Alone, they are non immunogenic. But when bound to any carrier molecule, they will become immunogenic. Like this was the case here. Like this antigen not necessarily induce the immune system because there are so many requirements for an antigen to elicit the immune response. Like there is a requirement for size, for molecular weight, and some other requirements are there for the antigens. If they do not fulfill those requirements, it will not generate the immune response. That I will cover in the next week. But heptanes can help an antigen to generate an immune response, like it can bind and form the heptane carrier conjugate, which will activate your immune system. So these are heptanes. These are small, non-immunogenic molecules. So this is the case here, like small molecule heptanes on conjugation with the immunogenic carrier. Like when these heptanes were injected into the any animal model, and then there was no production of this antiserum, like no immune system was activated, no antibody was produced. But when these heptanes were cross-linked with some immunogenic carriers and then injected into the this animal model, it produced the antiserum. It gave the absorbance readings for the antiserum, that is, it produced the antibodies. It means it activated the immune system here, but not in this case. Alone, heptane cannot activate the immune system. So this is the case here. So I'll explain this in the next class in detail. Don't worry. So let's quickly take the week 7 assignment. So I hope you have submitted this. So the first question was select all correct statements regarding the C3 convertase in the alternative classical and lectin pathways. So I hope now this is very much clear and there is no confusion with that. So quickly discuss all the options which are correct statements regarding the C3 convertase in all the three paths.
Yes, ma'am. C3 convertase in alternative pathway does not consist of C4B2A,
Yes.
uh, and it is present in classical pathway. So properdin stabilizes C3 convertase. Yes, it does in alternative pathway. In classical pathway, C3 convertase is formed by this complex, and C3 activity leads to generation of C5 convertase. Yes, it does. And in the lectin pathways, C3 convertase is identical to that of the classical pathway. Yes.
Yes.
And MASP2 is essential for C3 convertase formation in alternative pathway. No, it is essential for the formation in lectin pathway.
Lectin pathway.
C3 convertase is stabilized by C3b in classical pathway. No. And C3
C3A generated by C3 convertase acts on opsonil. No, C3B does, I think.
Yes. The B fragment was responsible for the opsonization, whereas the C3A or the A smaller fragments were responsible for the degranulation that we have seen here in
Yes.
this somewhere A and B, this here.
Ah,
Like this A
anaphylatoxin.
subunits were responsible for anaphylatoxin, that is the degranulation and the triggering of your inflammatory response, whereas this B subunits were responsible for your opsonization. So means your complement pathways are very much clear now.
Yes, ma'am.
Okay. So the next question was, what is the significance of the enzyme C5 convertase in the classical complement pathway?
It initiates membrane attack complex
It is
operation.
for the C5 convertase. Like it initiates the MAC formation. After initiation, it will bind with C6, 7, 8, 9. That will ultimately form a membrane attack complex. So the C5 have no point of going and binding to C2, C3, anything, and it will not even bind to this antibody. It comes at very later stage and it stabilizes C1Q1 antigen antibody complex. Now it will not go and interfere with any of the upstream molecules or upstream these fragments. So it initiates the MAC formation. So the next question was, which antibody type is most effective in initiating the classical complement pathway? We have seen this
IgM.
IgM. In the structure also, like you have seen this here, IgM is your pentamer. It is
Pentamer.
your five subunits and IgG your single subunit. So it is larger molecule, larger antibody. So it initiates your classical complement pathway. The next question is, which of the following best describes the role of heptane in immunology? Just we have seen this.
Yes, ma'am. It induces antibody production only when it is conjugated with a carrier molecule or a carrier protein.
Yes. A heptane can independently stimulate antibody production? No.
No, it cannot.
A heptane lack both foreignness and immunogenicity? No, it can be immunogenic when bound to a carrier molecule. A heptane directly induces immune response in animals without modification? No. After the modification only it can elicit your immune response. Heptane is structurally identical to an immunogen but has lower molecular weight. No, it is not identical. It is very small size molecule. So this is the heptane case. Next is which disease is primarily associated with the deficiency in C1 inhibitor?
Hereditary
Hereditary
angioedema.
angioedema that we have seen life-threatening inflammation occurs in this case because of the deficiency of your C1 inhibitor. Why is IgM considered more potent than IgG in activating the complement system? Why?
Because a single molecule of IgM can activate the complement system, whereas thousands of molecules of IgG is required. Minimum two molecules
Yes.
of IgG is required to activate it.
Yes. And nothing to do with the other options. It will not go and interact, bind with any of the convertase or any other immune cells. It have nothing to do with that. So directly, the answer is a single IgM molecule can activate the complement system, whereas multiple IgG molecules are required. So next question was, what is the role of C3 in alternative and lectin pathway? They are asking about alternative and lectin pathways.
It is hydrolyzed in form C3 convertase that leads to further cascade of reaction.
Yes. It will not directly bind to any pathogen surface. It binds to antibodies. No. It regulates the lectin pathway by interacting with the mannose binding lectin. No, it will not bind there. It neutralizes pathogens without cleavage. What options are these? So the correct answer is it is hydrolyzed and forms C3 convertase, leading to the amplification of the complement response. So the next question is, which cells are known as professional antigen presenting cells? We have been studying
Macrophages and
this.
dendritic cells.
Yes, this we have been studying since very initial lectures, macrophages and dendritic cells. Next is what is the key benefit of the alternative pathways amplification loop?
It quickly increases the deposition of C3b on the pathogen surface, enhancing opsonization and pathogen destruction.
Yes, the alternative pathways amplification loop. It will quickly increase the deposition of C3b
C3.
on the pathogen surface, enhancing the opsonization and pathogen destruction. What about the other options of this?
Like it is not involved in the recruitment of T cells initially, and then it enhances antibody production during no, it does not, and it leads to direct no, it does not lead to direct neutralization of viruses. It decreases the risk of no, it does not downregulate any inflammation.
It is not involved in the autoimmune response or anything. So the direct correct answer is it increased the deposition of C3b on pathogen surface. The last question was which of the following steps are involved in the activation of the classical complement pathway?
Yes, binding of IgM uh, C1q binding to CH2 domain and activation of C1 cleavage, and then recruitment of C4b and C2a, and that leads to the formation of C3 convertase, and then C5 convertase is being formed.
So this is the classical complement pathway. I hope now it looks much simpler when you know what is exactly happening there. Otherwise, before studying, it looks like so much complicated, like which factor will come after which one, who will bind with whom, what will be their action. It looks much complicated, but when you have learned this pathway, you know what are their role and what is their ultimate aim or ultimate goal. Like here, this C3 convertase will form. Here, the C5 convertase will form. When you know these things, then it is much simpler. And also, if someone asks you what is involved in the C3 convertase, then you should be knowing that how the C3 convertase form, like by the binding of the C4b and C2a, C3 convertase is formed. And what is the composition of your this C5 convertase? That is your C4b, C2a, and C3b. Like you should be knowing this. And after only the C5 convertase alone is not responsible for forming your MAC. It will bind to your C6, 7, your 8, 9. Like in MAC, this is involved. C5b, 6, 7, 8. This is involved, and poly C9 is involved. So this will form your MAC and it will ultimately kill your pathogen. So this you should be knowing very clearly. So these were the 10 questions of your week 7 assignment. And till now, the topics covered on the NPTEL portal are until your week 9 is released there. So you can check it out there and go through that. If you have any doubt from today's class or if you have any doubt or suggestion, any question, so we can discuss this.
Yes, I'm sorry, my connection was very poor right now. It
No, no
went
problem.
out.
No problem. It's fine. If you have any doubt or any question, you can ask.
No, ma'am, it's fine. Everything is clear.
Okay. And for the antigen and antibody structure function that we will discuss in the week 8 assignment, along with the week 8 assignment, we will cover this
Okay,
thing
ma'am.
there.
Yes, ma'am.
Till here, everything is clear. Or if from the previous week, if anything is not clear, then also you can let me know. I can cover that in the remaining time. I can prepare that for the next week. If anything is pending from the previous week.
Yes, ma'am, sure. I'll let you know.
Okay. So if you have
Yes,
any
ma'am.
doubt, any comment or suggestion, you can post in the discussion forum, or you can drop me an email, or you can also message me or email me. So do check it out this video from the YouTube channel, and see you in the next week. And I hope everyone have had a very colorful festival yesterday. So enjoy your festival and also keep learning.
Yes. Thank you, ma'am.
Okay, thank you. So if you don't have any doubt, you can log out.
Okay. Thank you.
Okay, thank you.